Assalamu alaikum guys. Okay, so today we're gonna start off with A level biology, CAIs. Okay, we're starting off with AS biology. We will, and by the way, I'm Dr. Shah Fahad. I'm an MBBS doctor, by the way, and I will be helping you out through your concepts of biology. Alright, so let's get started. This is Biology May June 2025 paper 1. Okay, I'm gonna pick up some random questions from here and try to simplify the concepts as much as possible. Let's start off with question number 1, which states that which statement about light microscope are correct. Option number 1 says to calculate the magnification of a light microscope, the eyepiece and the objective lens magnifications are added together. This statement isn't correct because they are multiplied, they're not added. The eyepiece, which is the the lens where you put your eye, and objective is the second lens that is at the bottom just above the stage. So objective and eyepiece. Objective generally has a magnification of 40 times. Eyepiece has a magnification of around 10 times. 10 multiplied by 40 is 400. That's how you calculate the magnification of a light microscope, not by just simply adding them together. So option 1 won't be considered correct. Option 2, the resolution of a light microscope is limited by the wavelength of light. That is correct because resolution generally, the smaller the wavelength, the better the resolution. But because light doesn't have a wavelength very small, it doesn't have a wavelength small enough to be comparable to the size of the components of the cell. So a lot of the components can't be visualized with the light microscope. So yeah, wavelength of light is quite big. The minimum wavelength that light can offer is 400 nanometers. So about 200 nanometers is the maximum resolution a light microscope can offer. Then the third statement, so option 2 would be considered correct. So it's gonna be option C then, 2 only, I guess. Let's look at the third one. The divisions on the scale on a stage micrometer are close together. The divisions on the scale on an eyepiece graticule. The divisions are not closer on a stage micrometer than an eyepiece graticule. No. The stage micrometer divisions are much bigger than the eyepiece graticule one. Eyepiece graticule is a lot closer. It has lines very next to each other, whereas the stage micrometer doesn't really have them that close. They're very far away. So you generally calculate the magnification using them. Alright, so that was the concept of light microscope. It's recommended for you guys to read about it from the recommended textbooks like Mary Jones. It's This chapter is very well summarized over there. Okay guys, so yeah, that's it for this lecture. Thank you so much. Inshallah, see you in the next lesson. Thank you.